Hi there, biology. Today we're going to have a look at cystic fibrosis. There's a couple of things that we need to learn how to do. The first one is to describe regulation of mucus water content in healthy lungs and also in lungs affected by cystic fibrosis. And the second one is to explain the effect of cystic fibrosis on the respiratory system, such as your lungs, digestive system, and your reproductive system. So let's start by just thinking about what we know about cystic fibrosis. Pause and make a few notes. Now you've made your notes, I would like you to go onto Moodle, download an article that I've put for you from the Biological Sciences Review magazine. We have 5 questions that we need to answer. The first one is, how are healthy lungs adapted for efficient gas exchange? Question 2 is, what is the name of the protein affected? Question 3 is, what is this protein responsible for? Question 4 is, what is mucus of a healthy person made of? And question 5 is, how is the respiratory system of a person with cystic fibrosis different? Have a read, answer your questions. And let's look through our answers. Firstly, in healthy lungs, we have a very thin barrier between the blood and the air, and also your lungs would have a very vast surface area, huge surface area. The protein that is affected is called the cystic fibrosis transmembrane conductance regulator, and thankfully we can shorten that down to CFTR. CFTR proteins are responsible for controlling the movement of chloride and sodium ions across cell membranes, and this is going to regulate the movement of water. In a healthy person, mucus is made out of water, ions, proteins, and sugar-coated mucins. We've got a longer answer for the next one. The respiratory system of a person with cystic fibrosis will have a dehydrated cell surface due to that faulty CFTR protein. Its cilia will not be able to beat properly. Its mucus is going to be more viscous, and this is going to have problems. This will block airways. It will make an environment for bacteria to grow. And due to the bacterial growth, this might cause some inflammation, where the airway walls are going to thicken and narrow and have the nasty side effect of producing even more mucus, making the original problem worse. So when we're looking at the biology behind this disease, we need to be happy with a few of our basic biological concepts. So this is mostly related to transport across cell membranes, and also it's going to be about basic things about how ions might interact. So we have a couple of questions for you to just sort of poke back into your memories and see what you know. The first one is, a cell has a lower concentration of salt inside than the surrounding solution. How will the salt molecules move? The second question is, sodium ions are positively charged. How might they interact with negatively charged chloride ions? The last question is that 2 areas are divided by a semi-permeable membrane. If an area has a higher salt concentration than another, how will osmosis be affected? Shall we look through our answers? In the first one, we can picture our cell in its solution. Our question states that there's a lower concentration of salt inside it, and there's a higher concentration of salt outside it. So clearly, the salt is going to move from an area of higher concentration to an area of lower concentration down the concentration gradient. In our second question, we have sodium ions are positive, chloride ions are negative. Hopefully that we know that molecules of opposite charges are going to be attracted to each other. This means they might move towards each other. In our third question, if an area has a higher salt concentration than another, we can think about water moving from an area of a higher water potential to an area of lower water potential through a semi-permeable membrane down the water potential gradient. Let's break this down into different pieces. My water potential describes an area where there are few water molecules and many solute molecules. So looking at the area inside the cell, you notice 1, 2, 3, 4 different salt molecules compared to this one salt molecule on the outside of the solution. So here we're going to have water moving inwards. So our blue dots representing the water, we have a higher water potential moving to this area of lower water potential. Our semi-permeable membrane here is going to be the cell membrane, And because we're going from high to low, we can describe this as down the water potential gradient. We can look at this in a bit more of a complicated situation where there are multiple areas that might have a higher or lower water potential, but we're going to do the same thing. The first thing, to identify the area of higher water potential, we want to see more solute and less water. So looking at the bottom here, we can see lots and lots of salt and not much water, so this must be the area of the lower water potential. Looking for an area of higher water potential, again, we've got more water here and less salt, so that means the first place that we might have things moving from, at the top here where we've got loads of water but no salt at all, will be from the top to the middle. So I've got my highest water potential on the outside, it's going to be lowest at the bottom, which means that we're going to have two phases of osmosis, first into the cell at the top and out of the cell at the bottom. Next up, we need to look at the structures that are going to be involved in cystic fibrosis. And here is a section of epithelial tissue. In this case, we have ciliated columnar epithelium, named after these little bits on the top. So what I'd like you to do is pause, and I'd like you to find out what these different boxes should be labelled as. Give yourself a moment. Let's look through our labels. This top surface we call the apical membrane. This bottom surface we call the basal membrane. And I just want to be very clear to everyone that we're not talking about the whole area of the cell. I'm literally just talking about the plasma membrane that lines this little bit at the top that contains all the cilia. That is the apical membrane. Again, at the basal membrane, I'm talking about the very thin cell membrane down here. That's the basal. Looking at my big label on the side, all of this layer is the epithelium. So the whole part of this bottom bit of the cell to the top bit of the cell, that's my epithelium. Underneath that I have connective tissue, which you can see contains fat, it contains nerves and capillaries. Let's start putting this together now. We've talked about separate concepts for a lot of different things. Now we have to assemble it. So we're going to start with a really rough summary of the issues in cystic fibrosis. This is really terrible. It doesn't have any keywords in it all. It's just so you can get an idea of what's happening. First, we want to think about our epithelial cell that you can see here, and you have the mucus lining on the top surface, on the apical surface. And we can think about it being too wet to begin with. So in a healthy person, here's what happens. You're going to start pulling out some of the sodium from the bottom. And I know that our problem is to do with water, so it seems strange to talk about salt, but we have great pumps that work for sodium and not so great pumps that work for water. So moving your salts around is going to be a much more effective way of drawing water out of an area that's too wet. So we start by losing sodium from the bottom. This is going to start pulling some sodium from the top. And this is going to create a concentration gradient where we have an electrochemical charge because sodium is positively charged and chlorides are negatively charged. So the chloride is going to get drawn down to the same place where we have this positive pool of ions. So all of these negative chlorides are going to come down, chlorides, sorry. But we know about salt and how salt affects water. So if I have a very salty area in the bottom here, that's going to leak some water out by osmosis. We're going to draw some water out this way. Next, we've got the water potential gradient in place. We're going to pull some water from the top. So through all of this process, most of it has just been about making a big pool of salt at the bottom that's going to draw water from the top of the cell right the way down, and this is going to make the water balance in the mucus fix itself. So again, that was very fluffy, no keywords really at all. Let's look at the full process. For this example, I'd like you to label it along together with me, and the next one you need to do by yourself. Now immediately you can see there's a lot of different structures here that weren't before. Let's set up what the different areas are. So at the top here, instead of drawing that sort of blue puddle, we know that we have the mucus. At the bottom here of the cell, we have the tissue fluid. We've got the top of the cell as this part, and we've labelled that before as the apical membrane. And the apical membrane has a few protein channels in there that are going to allow different molecules to move through. Same for the basal membrane at the bottom. These big purple blobs represent we have a different type of protein channel that's going to allow a certain molecule to move through it. So let's go through our first step then. The first step is about sodium, and we're starting with drawing the sodium at the bottom. The difference this time is that we have to put energy into this process. We're going to actively pump sodium molecules one at a time through the pump until we have a nice pool of sodium ions at the bottom here. And because we know about our concentration gradients, now inside the cell I can see there's very little to no sodium, so there's a lower concentration compared to the outside. Now we need to introduce a name for this channel here. This is called the epithelial sodium ion channel, or the ENaC protein. And what sodium can do is move through that protein to enter the cell. Because remember, we had none here before, so it's going to be drawn down by the concentration gradient through facilitated diffusion. Now we know we have this big pool of positive ions here, we've made an electrical gradient. Imagine these was just the plus charges, so we have 4 plus charges here and only 1 negative charge. This is going to draw down or attract those chloride ions that are at the top there at the moment, because we know positive things are going to attract negative things. So chloride ions diffuse from the mucus to the tissue fluid at the bottom through those little gaps between the cells. Let's look at what we've got here. I have in my tissue fluid, there are lots of sodium ions, there are lots of chloride ions, so we have this area where there's a lot of salt. Now I know about the salt concentrations, I can start talking about the effect it will have on water potentials. So where we have a lot of solute, we've got a lot of these salts, we're going to lower the water potential in the tissue fluid. That means that the tissue fluid is a lower water potential than the water potential in the cell. So, water is going to be drawn out of the cell by osmosis. And after the water's moved out, you can see now the cell has an area of lower water potential because it's lost the water that was in it before. So compared to the mucus, which is an area where there's more water molecules, so it has a higher water potential, the water is going to finally leave the mucus and enter the cell. So there is a lot of different things going on here. But as we said before, we want to pump salt out the bottom to diffuse salt in through the top. Next, we're going to move those chloride ions down to the bottom in the tissue fluid. Then we've got our water potential gradient, so I can pull water out the bottom, which makes a different water potential gradient here that will pull water out the top. And we can use all these lovely keywords to describe this. So let's put this into practice. I'm going to give you 6 diagrams to put in the right order. You just need to look and see, you can use the video to check along which phases should go where. After you've done that, I would like you to label a few things about each diagram to tell me which ion is going to be moving, where is it going from and to, which membrane is it going to go, or is it going between cells. I want you to write down the transport process, and as a challenge task, can you tell me why it is moving? What is the purpose of that movement of molecule? So give it a good go. Remember, you can look back through your notes from before, you can watch this previous part again if you need to see the process one more time, and then we'll go through our answers. Okay. So here we go, here's our first picture. So we start off with some sodium ions inside our epithelial cell, lots of sodium and chloride in the mucus, but very little at the bottom in that tissue fluid because the process hasn't begun yet. The next one that we should have has a couple more sodium ions here in the tissue fluid. And following that we should have the picture where we're starting to bring some of those sodium ions in from the mucus into our epithelial cell. After this, you should have a picture where we've got most of the chlorides and the sodium ions at the bottom. After this, what's changed is that instead of having water inside the epithelial cell, now it's in the tissue fluid with that area of salt. And then finally we finish with the mucus. You can see now there's very few ions in there, and there's also no water because that has just been drawn inside of our epithelial cell. So let's see about those descriptions. We're going to start by saying sodium moves across the basal membrane by active transport from the cell to the tissue fluid. Following this, we can talk about this area here, where sodium has moved across the apical membrane at the top by facilitated diffusion from the mucus into the main part of the cell. Next, over here, we have chloride, and it's moved through the gaps between cells by the process of diffusion from the mucus all the way through to the tissue fluid. After this, we can discuss our water movement by saying water moves across the basal membrane by osmosis from the cell to the tissue fluid. Finally, we can say water moves across the apical membrane at the top by osmosis again from the mucus to the cell. Mark yourself out of 6, please. See how many you got right. We need to pause for a moment to put all of that information together and see how many different things you can remember without looking at your notes. So hide everything away, and what I want you to do is describe the role of sodium in regulating excess water in mucus. So it's just the diagram we've talked about so far. See what you can remember. Okay, let's look through our answer. So we're starting at the bottom. We say the sodium pump on the basal membrane actively transports sodium into the tissue fluid. This is going to lower the concentration of sodium ions in the cell. The effect is that sodium ions will move by facilitated diffusion through the ENaC proteins from the mucus across the apical membrane into the cell. And this is happening from a high concentration to a low concentration. Moving the sodium then sets up an electrochemical gradient, which is going to attract the negatively charged chloride ions to diffuse between the cells from the mucus at the top to the tissue fluid. The end effect is that we have a water potential gradient formed between the mucus, the cell, and the tissue fluid. So score yourself out of 1, 2, 3, 4, 5, 6, 7 different marks. Add any corrections that you need, highlight any keywords that you've forgotten, because there's a lot of processes involved here, so you need to be really clear on what each of them do. When you're ready, we can look at the opposite scenario, where your lungs have too little water in the mucus, and you can see on that picture the arrows are now going the opposite direction. They're labelled 1 to 5. And what I'd like you to do is pause here and see if you can label what's happening. Use what you know about the different protein channels or protein pumps to explain what moves first by what process and where our concentration gradients are set up. Let's look through. So our label number 1 is about that chloride ion, and the chloride ion is going to get pumped into the cell this time across the basal membrane. This is going to cause chloride to diffuse through the open CFTR channels at the top. In the third step, sodium diffuses down the electrical gradient into the mucus, the electrochemical gradient, so you can see it going all the way from the basal membrane up to the mucus layer. And then the elevated salt concentration in the mucus is going to draw water out of the cell by osmosis. Lastly, in step 5, water is drawn into the cell by osmosis. So instead of having a sodium gradient pushing down this way from the mucus to the tissue fluid, we have a very similar process happening going upwards, where we start off with a pump. We have facilitated diffusion occurring, we have the attraction of the oppositely charged molecule, we have the water moving down the water potential gradient into the mucus, and we also have the water entering the cell after that, following the osmotic gradient, following the water potential gradient from the tissue fluid into the cell. But we need to think here of what would happen if this process broke. What would happen if the CFTR channel was not working? So if this channel that would let facilitated diffusion occur is not working, but the ENaC channel that allows sodium to move is always open. So have a look, have a think about what would happen. In order to do this, we need to have a few questions. So, here's our 5 questions that we need to work out. How are chloride ions transported into the cell from the tissue fluid? How are chloride ions transported from the cell into the mucus? Where is sodium moving from and to, and how is it moving there? And our last question is, why is water moving into the mucus? Have a go at answering, please. Let's have a look at our answers for the first one. So first off, chloride ions are transported from the cells to the tissue fluid through active transport. They're actively pumped from a low concentration to a high concentration. Our second question, how are chloride ions transported from the cell into the mucus? This occurs through facilitated diffusion from a high concentration in the cell to a low concentration in the mucus. When we think about where sodium's moving to and from, sodium ions will be moving from the tissue fluid into the mucus, and it's moving there for question 4 between cells through diffusion. So lastly, why is water moving into the mucus? We can say there is a low water potential in the mucus due to the presence of sodium and chloride ions. So water moves from an area of high water potential, such as a cell, into the mucus. So we've had 2 healthy scenarios. We've had a healthy scenario for too much water in the mucus and a healthy scenario for too little water in the mucus. Cystic fibrosis, remember, I asked you to think about what would happen when this protein channel is broken. So we're going to start off by saying there's a genetic mutation that causes the CFTR protein to be non-functional. But also the sodium channel is going to always be open. And unfortunately, this has the effect of always removing water from the mucus by osmosis. So, I would like you to take a moment to look at this big old table and write down some summary points about what happens when we're healthy but there's too much water in the mucus, when we're healthy but there's too little water in the mucus, and in the case of cystic fibrosis, when we have the CFTR channel is broken and the ENaC channel is wide open. Let's go through. So, our sodium ions start by being pumped out and they diffuse from the cell into the mucus. Next, we have chloride ions diffusing between the cells due to the electrical gradient from the mucus to the tissue fluid. Our water is moving out of the cell into the tissue fluid, and it's also going to move into the cell from the mucus. Our CFTR channel protein is closed. We needed to move the sodium ions from the top to the bottom. We didn't need to move the chloride ions upwards by facilitated diffusion, so this channel is shut. The ENaC channel is going to be open because this is what's allowing those sodium ions to enter from the mucus. That's what's allowing them to diffuse into the cell from the apical membrane. On the bottom, we have the sodium pump we know is working because it needs to do this step, pumping the ions out the cell into the tissue fluid. And the chloride pump is closed because remember, we're not moving the chloride ions here. So the mucus that started out being too wet is going to become dry and the water is drawn out. So next up, when we have this healthy person with too little water in the mucus, remember we needed to move some things from the bottom up to the top in order to draw water that way too. So sodium ions in this case weren't being pumped. These were the ones that are diffusing between that little gap between cells due to the electrical gradient. So this goes all the way from the tissue fluid at the bottom up to where the mucus layer is at the top. Next up, with our chloride ions, these are the ones that are going to be pumped at the bottom of the cell upwards first. And then they're going to be diffusing from inside the cell up through the apical membrane through the CFTR protein. So water's moving in 2 steps. First one is going to move out of the cell into the mucus through the top, and it's also going to move out of the tissue fluid into the cell. And it's doing this because we've built up this very salty chloride area first, and then we're going to add these sodium ions to it, so you have a big pool of salt at the top this time, and water will follow through osmosis. It's going to move to the area of low water potential. So our CFTR channel, we know that's involved in the facilitated diffusion of the chloride ions, so that's going to be wide open. And the ENaC channel is going to be closed because we are not diffusing those sodium ions through facilitated diffusion. They're moving through the gaps between the cells. Our sodium pump similarly is going to be closed because we didn't start our step by pumping the sodium ions out the bottom. We started by pumping the chloride ions up into the cell, which means that our next one is, yes, it is open. It's used to pump those chloride ions from the tissue fluid into our cell. And our mucus, we can look over at our water column to get ideas for this. It's going to become more watery. Water has been added from the contents of the cell. So let's compare this to cystic fibrosis. In cystic fibrosis, ions are going to be diffusing from the mucus into the cell. And sodium ions are also going to be pumped out of the cell into the tissue fluid. So this is much like when we have a healthy person with excess water in the mucus. The chloride ions are going to diffuse between the gaps due to that electrochemical gradient. So we're going from the top where there's the mucus into the tissue fluid. Our CFTR channel, I'm jumping ahead. The water is going to move out of the cell into the tissue fluid, so you can see these three steps and these three steps are identical. The response in a person with cystic fibrosis is very similar so far to a healthy person with excess water. Our CFTR channel is broken. Our ENaC channel is permanently open. The sodium pump is working, just as in a healthy person with excess water, to pump sodium ions into the tissue fluid. The chloride pump is closed, but the issue is, this is a response only when we have too much water. In cystic fibrosis, regardless of water levels, we're continually drawing that water out of the mucus layer into the cell and into the tissue fluid. So instead of resuming the normal water balance of the mucus, what happens is that the mucus becomes very dry and very viscous or gluey and thick. So again, this is a lot of information. We need to take a pause point to bring some of these things together. I would like you to explain how the expression of a gene mutation in a person with cystic fibrosis causes a buildup of mucus in their respiratory system. If you're feeling confident, go ahead. Otherwise, I have a few more things I could ask you. So if you're still with me on this, the first thing we need to think about is why they've even mentioned there's a gene mutation. We know that genes code for proteins. So can you think of a protein channel that is going to be affected by this disease? So think about the structure of your cell. We have a few different pumps that are involved. One of them is going to be always open, and that causes some problems. One of them is going to be always closed, and that causes some problems. So think, what is the name of that channel that's always closed? And what is the name of that channel that stays open? The next one is that we need to think about this buildup of mucus in their respiratory system. So we know that mucus is going to be on this surface. You might be able to use a keyword to describe what surface that is. You might be able to tell me a few things that happen at the bottom here that are going to cause some trouble. Something else that's going to be drawn down from the top. You might be able to tell me what the condition of this mucus is like. And how is that going to affect their respiratory system? So what organs are in that respiratory system? You have 5 marks. Give it a go. Now, I'm not going to go through the answers of this one with you. I would like to take it in on your Moodle submission and mark it myself. So, that's the end of this topic for now, until you get your feedback for the questions you'll submit in your booklets. The next step, and warning, there's a few quite grim pictures coming up about how your organs are affected by cystic fibrosis. So the first thing that we want to do is look at the lungs. So there's two pictures here of lungs that could have been infected by cystic fibrosis. And you notice if you have this dark picture on the x-ray, that would be showing you where we have healthy lung tissue. But where we have all of this white tissue here, that shows that we have unhealthy lungs affected by cystic fibrosis. Same on this side, these are not happy, healthy looking lungs. There's a lot of mucus and infection rife. So what we can look at first is a diagram of these lungs. And I would like you to label this picture first in your diagrams, and then we're going to talk about how cystic fibrosis affects them. Alrighty, let's look through our labels. We start off with the trachea at the top, and we have our rib bones shown there. In between them it's called rib muscle, but we can be a bit fancier than that and call it intercostal muscles. We have the bronchus, or bronchi if you're talking about two of them, followed by bronchioles, which are smaller divisions of the bronchi, and at the end of our bronchioles we have little alveoli. Underneath this we've got a few membranes we need to know about. So obviously the heart is not part of the lungs, and respiratory system is just kind of there because that's where it be. Underneath that we have the lungs and the diaphragm. Let's look through our process. So starting at the top, the mucus is going to become more viscous in cystic fibrosis. We know that lungs and their epithelium are aligned with epithelial cells that have cilia on. So if I draw you a little picture of what that looks like, imagine you've got that cell that we talked about earlier, and it's going to have really fine hair-like projections at the top. And these are going to wiggle around, and they can help move mucus normally. So that would go all the way up to that junction at the top where you can move things into your esophagus, and it would be able to tip that excess mucus from your trachea down into your esophagus to be digested. And that's how we can get rid of a lot of mucus with trapped dust and things. So this time, cilia can't move it, it's just going to stay there being stodgy and unpleasant. And unfortunately, this means that the bacteria are going to get trapped in the mucus, and it makes a place for them to grow, so we get bacterial infections. Because we also have a build-up of viscous, sticky mucus, it's going to block up the airways of a person, and this means that less oxygen is going to be able to flow through the trachea, down into these areas, and then finally to the alveoli at the end. If you imagine having a lot of mucus blocking those channels up, it makes this a bit more difficult for your person to get their air containing the oxygen, which has the effect of them getting less oxygen flowing to the alveoli. If there's less oxygen, there's going to be a reduced concentration gradient for it, and ultimately this means that your alveoli will have reduced gas exchange. Your lungs are working less effectively due to all the mucus that are blocked up there. Next we're going to have a look at a pancreas. So here we've got a healthy pancreas and we have a not so healthy pancreas up top. Let's have a look at what's affected here. First off, the sticky mucus is going to block this tiny little tube, the little pancreatic duct, that is going to allow the digestive enzymes from your pancreas to travel through it and go and be deposited into your digestive system, into your intestines. But this can't happen. If we have this blockage here that you could see before, you notice that that's blocking up this area, meaning that the digestive enzymes can't get through. Think of what the enzymes might do in your intestines. This means the food is not digested, and if food isn't digested and broken down, you can't absorb it, so fewer nutrients enter your body. But we still have this problem, not just that we're not digesting our food, you have a blockage and that's going to cause a build-up, right? So the trapped digestive enzymes are going to be damaging the cells around the area where they're trapped, and this means that the cells can't produce insulin. Next up, we're looking at the reproductive systems, and you notice here we're looking at a male rat that's been dissected, and the vas deferens is this tube connecting to the testes here. And you notice in a wild type, so an unaffected person, you have two very clear vas deferens, but they are not so developed in the rat that has the mutation for CFTR protein, where it's broken. And the reason that this happens is for two reasons. If you're a female and you don't have testes or a vas deferens, the mucus plug is going to develop in the cervix, which means that you cannot have fertilization occurring. You have a reduced chance of pregnancy because sperm are not going to be able to pass through and enter and reach the egg, which usually will be up here more towards the fallopian tubes. Unfortunately, in males, the vas deferens is completely missing, or it will be underdeveloped. Next up we have cystic fibrosis and sweat. So there's a few things that can happen here. In a healthy person, you might release some of these sodium and chloride ions from your body to your skin, and we know that water is going to follow. It's going to go down the water potential gradient by osmosis, and it will enter the surface of your skin. This means that the water can evaporate to cool off your body, and then afterwards you reabsorb your sodium and your chloride. So if you think about it, we have the salt layer first, water is drawn up afterwards, water evaporates, and you take in your sodium and your chloride ions again. But we have an issue with cystic fibrosis. Those chloride ions are not reabsorbed because we have that broken protein. So in cystic fibrosis instead, if you have this pool of negatively charged chloride ions, you're going to draw more sodium ions out behind it due to that electrochemical gradient. That means that you're going to keep losing salt, and you can have symptoms such as fatigue, vomiting, and dehydration. So, to finish off our lesson, there are 3 questions in an exam style. I would like you to try and summarise the answers, please, from memory. Only use your notes as a last resort. And then submit them in your booklets to Moodle, and I will mark them for you. As always, if you have any questions, send me an email and I can try and redirect you to a few more sources or answer the question myself. Take care, people. See you later.